to Leiden to start a postdoc in protein-protein interactions. And then he was a bit in a search for a more biological problem, and he decided to come back to Portugal in 2004 with a Marie Curie reinsertion grant, and he started studying malaria. So it's just a proof that at any time, if you wish, you can change fields. So thank you very much for being here with us today, Miguel. And I know my connection is bad, but I'm going to stop now. So hopefully yours will be better than mine.
Okay, well, is everybody, can you hear me well?
Yeah?
Okay.
Yes.
So I would like to start obviously by thanking the organizers of this series of seminars for inviting me and for giving me the honor to be the first speaker in this new series of seminars. Particularly, I'd like to thank Professor Manuela Costa for this invitation. And like she was saying, I am a researcher at Instituto de Medicina Molecular working on malaria, and I have been working on this field for about 16 years now. I didn't start in this field like Manuela said, but I started working on this field quite a few years ago now, and I am now a group leader at IMM, and I have several projects in the field of malaria going on in the lab. But today I would like to bring to you a project that has been probably the one that has had the most visibility in the lab, and also the one that that has started when the lab started and has been going on for several years now, for more than 10 years now. So what I'm going to try to do is to summarize in 30 minutes, which is the time that I have, about 10 years of work. So as you can imagine, there's going to be a lot that will be said, but a lot that will be left unsaid. And I urge you to please interrupt me if you have any questions at any point. I always ask that you interrupt during the talk if you have questions, because then it is a problem if you are left with something that you didn't understand, and then it may prevent you from understanding everything else that comes after. So feel free to interrupt, or if you prefer, leave your questions until the end. And I'm going to start immediately, because otherwise I'm going to run out of time. I just want to ask Manuela or any of the organizers to please give me a hint if I'm starting to exceed my time. I've tried to make the presentation fit 30 minutes, but like I said, this is a lot of work condensed in 30 minutes, and I'll try to be as clear as possible, but stop me if you need any clarification. So the work that I'm going to be telling you about concerns the development of a new vaccination strategy against malaria. And malaria, as you likely know, is a very prevalent disease. It is one of the 3 deadliest human infectious diseases that out there, and it is a disease caused by Plasmodium parasites. So it is a parasitic disease. It is a parasitic disease caused by parasites of the genus Plasmodium. Sorry, I keep getting these notices that people are on the waiting room to admit them. Can somebody else admit the people into the room so that I can just focus on the presentation?
Yes, I will do that. Don't worry.
Okay, thank you. So it is caused by Plasmodium parasites, and as I think most of you probably know, it is an infection that is transmitted to humans via the bites of infected mosquitoes, Anopheles mosquitoes, as it turns out. And there are 5 species of human malaria parasites, but there are 2 that concern us the most. That's Plasmodium falciparum and Plasmodium vivax. These are the 2 species of human malaria parasites that cause the most mortality. In fact, Plasmodium falciparum causes by far the highest mortality, but Plasmodium vivax is a big problem because it is spread in many regions of the world, and it is a very big challenge to eradicate. So most of the mortality attributed to malaria occurs in sub-Saharan Africa, and it's mostly children under the age of 5 years old that succumb to malaria. And we are talking about more than 400,000 deaths every year attributed to this disease. So there is a parasite, there is a human host, there is a mosquito. How do these 3 entities come together? Well, the cycle of Plasmodium, the life cycle of Plasmodium parasites is complex, and it goes through different stages. And it is initiated when an infected mosquito injects parasites into the skin of the mammalian host, and these parasites go to the liver, where they infect hepatocytes and replicate inside these hepatocytes. We call this the pre-erythrocytic stage of infection because it precedes the erythrocytic stage of infection, where the parasites that are released from the liver will infect red blood cells and cause the symptoms of disease. It's important to note that the infection of the liver by Plasmodium is asymptomatic, so there are no symptoms of malaria associated with liver infection by Plasmodium, but it is an obligatory phase because only when the parasites exit the liver and reach the blood will they cause the disease symptoms. This cycle is completed when a mosquito ingests the sexual forms of the parasite from an infected host, and then they will generate new parasites inside the mosquito that are ready to be transmitted to another host. What I would like you to retain from this is that the pre-erythrocytic stage or liver stage of infection is an ideal target for prophylaxis and vaccination because of its obligatory nature and because of its asymptomatic nature. And it's important to just retain a couple of concepts here. One is that the parasite form that infects the liver cells is called the sporozoite. So the parasite goes through different stages with different shapes and forms, and each one has a name. The only name I need you to retain is sporozoite, because this is the form of the parasite that is injected by the mosquito and infects the liver cells. And these sporozoites will be at the core of our of the rest of the talk, because they are at the core of the vaccination strategy that I will be talking about. So we need a vaccine against malaria. Such a vaccine does not exist yet, contrary to sometimes people think that there is a vaccine against malaria. No, there is not a vaccine against malaria. And when we look at the cycle, the malaria cycle that you can see here represented in a slightly different way as before, but it's the same principle. Sporozoites infect liver cells, replicate inside liver cells, and parasites exit to the blood, cause blood infection and infect the liver and cause symptoms, and then they eventually go back to the mosquito for a new infection. And when we look at this cycle, we could think of 3 stages where a vaccine could act. At the blood stage, preventing infection of the erythrocytes; at the transmission stage, preventing transmission from one host to the other; or at the liver stage or the pre-erythrocytic stage before symptoms arise. And like I was saying, this is indeed the stage of the life cycle where vaccines are most successful. And we can distinguish between 2 types of vaccines that act on the liver stage of infection by Plasmodium: the subunit vaccines or the whole organism vaccines. Subunit vaccines, as the name indicates, are vaccines based on a single antigen or a fraction of that antigen that is used to expose that antigen to the immune system, and then the immune system mounts a response against it that is able to protect against infection by the parasite. And the most well-known subunit pre-erythrocytic malaria vaccine is RTSS. It is a vaccine based on one particular antigen of the parasite called the circumsporozoite protein, CS or CSP. And this protein is the most abundant protein on the surface of the sporozoites, and it's very immunogenic, and it makes it the major pre-erythrocytic vaccine target. So the RTSS vaccine is composed of a portion of the circumsporozoite protein that is then delivered in a viral vector form to generate immunity against Plasmodium. It is thought that, the data show that this vaccine acts mainly through the production of antibodies and not so much cellular immune responses. And this is the most advanced vaccine candidate against malaria there is. So it is currently under pilot studies in several countries in Africa, but there is a very big problem with this subunit vaccine. It's that its efficacy is modest. It's about 36% efficacy only, which is not what we would like to see for a truly effective vaccine against malaria. So it is the most advanced. It is close probably to being licensed. It is going to be licensed under the commercial name of Mosquirix, developed by GSK, but it is not the vaccine that we desire for malaria because of its low efficacy. So if this is a low efficacy vaccine, what alternatives do we have in order to get a more efficacious vaccine? Well, this is where whole sporozoite malaria vaccines come into play. Whole sporozoite or whole organism pre-erythrocytic malaria vaccines, as the name suggests, are based on the use of the sporozoite itself as the immunization agent. The idea is that you get your infectious parasite and attenuate it in some way. I'll show you how. And that this attenuated parasite can then be used as a vaccine against malaria. So instead of showing one antigen to the immune system, you are actually showing the entire complexity of the antigens that compose the parasite to the immune system. And then the immune system mounts a very powerful response against a new infection. And this is the only strategy that has consistently shown to convey sterile immunity against malaria. Sterile immunity means that there is a complete blockage of a new infection after the subject has been vaccinated. So whole sporozoite malaria vaccines or whole organism malaria vaccines are based on sporozoites. What you see on this picture here is a salivary gland of an infected mosquito full of sporozoites. There are thousands and thousands of sporozoites in the salivary gland of a single mosquito. And these sporozoites are our vaccine. They are the agent that is used to vaccinate. But of course, the common whole sporozoite vaccines that exist are based on Plasmodium falciparum, so the most infectious, the deadliest human malaria parasite. So this needs to be attenuated before it can be used as a vaccine. And there are several ways to do this. You can attenuate it by irradiation. You can genetically modify it so that it is incapable of completing its development in the liver, or you can even administer it without any attenuation, but kill it as soon as it reaches the blood. In all cases, what you have is that the parasite infects the liver cells and elicits an immune response that is then protective against a new infection. So to summarize, you have radiation attenuated sporozoites in which parasite irradiation leads the parasite to arrest its development in the liver. You have genetically attenuated sporozoites in which you have modified the parasite by deleting genes that are essential for its development in the liver. So it also arrests its developmental process inside the liver cells. And then you have non-attenuated parasites that are free to progress through the liver stage, but then they are killed as soon as they reach the blood by a circulating drug. All these strategies are based on Plasmodium falciparum parasites, and the data show that their efficacy depends largely on cellular immunity as opposed to humoral immunity, which is based on antibodies. I don't know exactly how much you know about immunology, You don't need a very big knowledge of immunology to follow this, but just keep in mind that broadly, there are two main kinds of immune responses, the humoral responses, which are antibody-based, and the cellular responses, which are cell-based, T-cell-based, lymphocyte-based. And in the case of these whole sporozoite vaccines, efficacy is largely dependent on cellular immune responses, mainly CD8 T cells and gamma delta T cells. But there is also a contribution of antibodies against the circumsporozoite protein, which is, like I said, the strongest immunogen in the parasite. So to summarize, current whole sporozoite malaria vaccination approaches rely on the use of Plasmodium falciparum sporozoites, which are deadly to humans. This raises possible safety concerns because you need to ensure 100% attenuation to prevent disease. They also do not protect against Plasmodium vivax, which is another human infectious parasite against which we need a vaccine. And then there are several limitations or possible drawbacks of each one of these alternative strategies to make a Plasmodium falciparum based whole sporozoite vaccine. But whole sporozoite vaccines are indeed the best bet we have for an efficacious vaccine against malaria, but Plasmodium falciparum based whole sporozoite vaccines have some limitations or potential limitations. And this is where our strategy for an alternative whole sporozoite vaccine against malaria comes into play. So what I said until now sort of sets the stage for the actual work that we have been doing and that I'm going to be telling you about from this point onwards. And to understand what we proposed, you need to go back to the origins of vaccination when Edward Jenner created the first vaccine that the world ever knew, which was the vaccine against smallpox. And he did this when he realized that people exposed to a bovine variant of smallpox, which is called cowpox, appeared to be immune against smallpox. And so he began inoculating people with cowpox, the cowpox virus, and realized that people inoculated with the cowpox virus were protected against the smallpox virus. So the inoculation of a relative of the human infectious virus was able to elicit immune responses against that virus. What does this have to do with malaria? Well, a lot, because also in malaria, we have Plasmodium parasites that infect humans, such as Plasmodium falciparum, but we also have Plasmodium parasites that infect other species, other mammalians without causing disease in humans. And the most well-studied example of such a parasite is Plasmodium berghei. Plasmodium berghei is employed in laboratories around the world for decades as a model to study malaria in the laboratory, and it's a rodent infective parasite that does not cause disease in humans. And so you can see here the idea that you start to see the idea that we proposed. The idea that we proposed is a little bit like Jenner used the cowpox virus to immunize against smallpox. We propose to use Plasmodium berghei to immunize against human infective Plasmodium species. And the idea is that there will be cross-species protective responses elicited by Plasmodium berghei that will be protective against Plasmodium falciparum. And this is not unlikely because there are a lot of CD8 T cell epitopes that are shared by the two parasites, the rodent Plasmodium berghei parasite and the human Plasmodium falciparum parasite. But on top of that, we proposed an added bonus to this vaccine, which is to modify the Plasmodium berghei parasite to insert antigens of the human infective parasite. It's like you put a coat of or a disguise or a mask of human parasite on your rodent parasite. And in this way, your rodent parasite becomes a chimeric parasite that also has a component of the human Plasmodium falciparum parasite. And in this way, our expectation is that we will get, on top of the cross-species immune responses because of the similarities between the two, we get targeted immune responses that are specifically elicited by the antigen of the human parasite that we genetically introduced on the Plasmodium berghei rodent parasite. And this is exactly what we did to create our vaccine candidate. Our vaccine candidate is called PbVac. And PbVac, you won't be surprised to hear, that is made of Plasmodium berghei, into which we inserted circumsporozoite protein, CS or CSP, of Plasmodium falciparum. I told you before, the circumsporozoite protein is the most abundant protein on the surface of sporozoites. It's the major immunogen in whole sporozoite vaccines. So it's no surprise that this is the antigen that we chose to insert into the Plasmodium berghei platform. So what we have now is a genetically modified Plasmodium berghei parasite that we call PbVac, and which expresses, besides its own range of antigens, we didn't remove anything from Plasmodium berghei, but we added the circumsporozoite protein of Plasmodium falciparum. So PbVac is a parasite that expresses, besides its own array of antigens, it expresses the Plasmodium falciparum circumsporozoite protein, or CS or CSP. And so we showed on the right here, you can see images that show that this chimeric parasite expresses both its endogenous CS and the inserted CS. You can see that sporozoites and liver stage parasites express both these proteins. So the genetic insertion worked, and the expression of this antigen works as well. And so now we have our vaccine candidate. The first thing we need to know, or that we had to do, this is already a few years ago, was to show that this Plasmodium berghei parasite that was genetically modified to express Plasmodium falciparum CS, so our PbVac parasite, would be able to infect human hepatocytes. Because, like I told you, whole sporozoite vaccines exert their activity, or they elicit the immunity when they infect the liver cells of their host. So we need our rodent, our modified rodent Plasmodium parasite, to be able to infect human hepatocytes. And we did this, we showed that this was the case in a number of ways. I don't have time to show you all the data. I'll just summarize it with a piece of data that was using liver humanized mice. These are mice that have been engineered to have human hepatocytes in their liver. And we showed unequivocally that Plasmodium berghei, or PbVac, could infect the human hepatic cells in these liver humanized mice. The other thing we needed to show was whether PbVac was unable to infect the human red blood cells, because, like I said, that's when the symptoms of malaria happen, and we didn't want that our vaccine would be able to infect and develop in the human red blood cells, because that would cause disease. And so we did this also in a number of ways, which I summarize in a single figure, in which we used blood humanized mice. So these are mice whose blood is replaced, not 100%, but about 90% of the erythrocytes in circulation in these mice are human erythrocytes, human red blood cells. And we used this model to show that PbVac is unable to replicate and develop inside the human red blood cells, and therefore it cannot cause disease. After this, we had a huge number of experiments that needed to be done before we could think or even dream of testing the vaccine in humans. And these experiments took several years with several animal models. And again, I don't have time to show you everything that we did, but I'll summarize it in this slide, which shows that we used PbVac to immunize different animal models, including rabbits, mice, and non-human primates. And consistently in all these preclinical models, what we observed was that immunization with PbVac elicited immune responses in all these animals. And this is very important, and I call your attention to this, please. There were always two types of responses. On the one hand, cellular immune responses, which we showed were mostly derived from the Plasmodium berghei scaffold of PbVac. So from the cross species immunity between Plasmodium berghei and Plasmodium falciparum. But then there were antibody responses, humoral responses in all these animal models that were totally or mostly dependent on the Plasmodium falciparum circumsporozoite protein that we inserted on PbVac. So just to summarize, immunization with PbVac elicits cellular and humoral immune responses. Cellular responses are mostly cross-species responses between Plasmodium berghei and Plasmodium falciparum, and the antibody responses are mostly directed at the Plasmodium falciparum CS that we inserted in the vaccine. And we showed that those antibodies against CS are functional in inhibiting an infection by Plasmodium falciparum. So with all these, and like I said, this is just summarizing many experiments, but when we completed these, we thought this is now time to go and test in the clinic in humans. But as you can imagine, this was not an easy task because there was no previous history of experimental administration of rodent malaria parasites to humans. There had been vaccine tests before in which humans were, people were injected with human infective parasites, but not with rodent malaria parasites. And on top of that, PbVac is a malaria parasite of rodent origin. It's a genetically modified organism, and it has to be produced in mosquitoes that feed on the blood of infected mice, because that's the only way to generate the sporozoites that constitute the vaccine. So you can imagine that when we started thinking about going into the clinic with this, it was really a lot of red flags coming up, saying there were a lot of people that told us you'll never be able to do it, you'll never get authorization to do this in humans because this is too far from anything that has ever been done. But we got the Malaria Vaccine Initiative, which is an entity that coordinates malaria vaccine development worldwide. We got them to work with us, or we worked with them, and with their advice and with a lot of input from various entities, we essentially did everything that we could to demonstrate that it would be safe to administer PbVac to people. Again, this is several years of work summarized in one slide. It's all published if you want to see the details. But basically, we had to create and validate a master cell bank of PbVac. We did all kinds of microbiological analysis to show that it was not contaminated with anything else. We did biodistribution studies, drug sensitivity studies, toxicology assays, basically everything that the authorities asked us to do in order to have some assurance that it would be safe to do this in humans. And so in the end, we did get authorization to perform a clinical evaluation of PbVac in humans, and we were able to test our vaccine in human subjects. This clinical trial took place in the Netherlands, and it went on in 2 phases. The vaccine was administered by mosquito bite. I know this always sounds strange when I say this, but for this kind of vaccine, a whole sporozoite vaccine, at this stage of clinical evaluation, it's the regular procedure to administer it by mosquito bite, because the sporozoite is the vaccine, and the sporozoite is made inside the mosquito, and it's in the salivary glands of the mosquito. So the administration of the vaccine was done by mosquito bite, and then the clinical trial proceeded in 2 phases. First, a phase 1 of safety, which is essentially to determine whether the vaccine is safe and well tolerated in volunteers. So what we did was we had groups of volunteers that received increasing numbers of the vaccine, so increasing numbers of mosquito bites with the vaccine. So one group received 5 mosquito bites. We then monitored the appearance of any adverse events. Nothing happened. Everything was okay. We moved on to 25 mosquito bites. Again, monitoring adverse events. There was nothing serious. We moved on to group 3, in which they received 75 mosquito bites. And then we determined that this administration did not have any severe adverse events. And so this group 3 moved to the phase 2 of the trial, which is already the efficacy trial. And here they received another 3 immunizations with 75 mosquito bites each. And then they were challenged with infectious Plasmodium falciparum sporozoites, with the actual infective parasite. Of course, there was a control group that was also challenged with the infective Plasmodium falciparum and was not vaccinated, because we needed to compare what happened in the controls versus the vaccinated or immunized subjects. And so what are the results of these clinical trials? So the phase 1, the safety assessment showed that there were no adverse events, no breakthrough infections, meaning that PbVac did not move past the liver into the blood of the human volunteers, which would have been very bad. So that did not happen. And there were no serious adverse events during any stage of this trial. Of course, there were local adverse events like this, because if you get 75 mosquito bites in one small area of your skin, of your arm, there will be a reaction. And of course, these are adverse events of the administration itself. It's not an adverse event of the vaccine, it's of the administration method. And yes, believe it or not, there were volunteers to do this, and we were able to do the entire assay with volunteers that didn't give up after the first immunization. And the group 3 volunteers actually went through this 4 times. And it looks scary, but it really disappears quite quickly. And the next day it's already okay. But yes, there were local adverse events, but no serious adverse events. So in terms of safety, we showed that it was safe. PbVac is safe and well tolerated. And then what happens in terms of efficacy? This is what really matters. We vaccinated a group of people. There was a group of controls. Both were exposed to Plasmodium falciparum, and now we need to know what happened in terms of the infection by Plasmodium falciparum. And this is where we have these results here that show what happened. So we were, on the one hand, a little bit disappointed, but on the other hand, very happy, because although none of the volunteers was completely protected against infection by Plasmodium falciparum, we had the appearance of Plasmodium falciparum parasites in the blood, so that went through the liver into the blood, was very significantly delayed in the vaccinated volunteers compared to the controls, as you can see in this graph. Also, the amount of parasites that appeared in the blood of the immunized volunteers was significantly lower than in the controls. Now, these 2 parameters here, the time that it takes for the parasite to appear in the blood and the density of parasite that appears in the blood, allow us to calculate what the efficacy of the vaccine was in terms of decreasing the infection of the liver by Plasmodium falciparum. And this calculation shows that we have a 95% decrease in liver parasite burden, in Plasmodium falciparum liver parasite burden in the immunized volunteers compared with the controls. Now, the question is this good or is this bad? Well, depends on how you want to look at it. 95%, you need to get 100% blockage of liver infection in order to prevent the symptomatic stage of the infection. So 95% is not enough to be protected, because you need to block completely the parasite in the liver. But 95% is very close to that goal. So in that sense, it gives us the hint that we can get to the complete protection we want by tweaking the parasite or by tweaking the immunization process, and I'll show you how. And so what we have is a vaccine that clearly had a very potent biological effect that decreased Plasmodium falciparum infection in the liver of the immunized volunteers by 95% relative to the controls. And then we tried to understand what was going on in these volunteers that could tell us how we could go from 95% to 100%. And so we did an array of immunological analysis of these volunteers. And again, I can't show you everything, but this is all published and you can look at it if you like. And so we looked at antibody responses to Plasmodium falciparum. We showed, so in all these graphs that I'm going to show, I1 means, I1 minus 1 means before, it's samples collected before the first immunization, and C minus 1 means samples collected after 4 immunizations before the challenge, before exposure to falciparum. So you can look at each volunteer, each number there is a different person, a different volunteer, and you can look at whatever analysis we are doing before and after immunization. And what you can see is that after immunization, you get antibodies in the serum in the blood of these volunteers that recognize Plasmodium falciparum sporozoites. Not only that, but more importantly, the serum, the antibodies in the serum of these volunteers are able to block the invasion of hepatic cells by Plasmodium falciparum in vitro by 61%. And if you deplete from this serum the antibodies specifically against PFCS, you lose that ability to block infection. So in summary, what these results show is that our Plasmodium falciparum sporozoites, which would normally infect human liver cells, in the presence of these antibodies against Plasmodium falciparum CS that were elicited by immunization, lose their ability to infect the human hepatic cells. Not completely, but to a significant extent. And so we showed that that immunization with PbVac elicits the production of antibodies against the circumsporozoite protein of Plasmodium falciparum, the PFCS, that we engineered to make PbVac, and that these antibodies are functional in blocking an infection by Plasmodium falciparum. And we further showed that the amount of the antibodies that we get is proportional to the number of mosquito bites that the volunteers received. So there is a dose-dependent amount of antibodies that is produced when you immunize with PbVac. So more vaccine means more antibodies. And this is already a hint that we can increase the efficacy of our vaccination by increasing the dose. I'll show you how in a minute. We also looked at cellular immune responses. We showed that both CD8 T cells and gamma delta T cells, their frequencies increased after immunization, which is good because we know that these are the cells that are normally associated with protection by whole sporozoite vaccines. And we showed that these cellular immune responses are functional because when you stimulate these lymphocytes with Plasmodium falciparum sporozoites, you get production of cytokines like interferon gamma, TNF alpha, or IL-2. And again, this is a dose-dependent effect. And we showed that this is dependent on Plasmodium berghei itself, the backbone. So Plasmodium falciparum CS contributes very little to this. This is dependent on the cross-species immune responses between berghei and falciparum. So it's totally in agreement with what we saw in the preclinical models. So PbVac has two components. On the one hand, there is the berghei parasite. Then we put Plasmodium falciparum circumsporozoite protein on top of it. And then immunizing with that, we get humoral antibody responses against Plasmodium falciparum CS, and we get cross-species cellular responses against Plasmodium falciparum. And so in summary, from our clinical data, we showed that administration of PbVac is safe and well tolerated. We didn't see sterile protection, but we saw a 95% decrease in the liver infection by Plasmodium falciparum in the immunized volunteers. And we showed that immunization with PbVac elicits antibodies against Plasmodium falciparum CS, which are functional in blocking infection, and also dose-dependent cross-species cellular immune responses. And so this is where we are, and where do we want to go? So I told you we are close to 100%, but we are not there yet, and we need to be at 100% blockage of liver infection for this vaccine to work. And so how can we do that? I told you that the vaccine was administered by mosquito bite, and the volunteers received 4 times 75 mosquito bites each in order to get these results. We cannot go higher than that. We cannot ask people to be bitten by 1,000 mosquitoes in order to increase the dose of vaccine. So what can we do in order to increase the dose of vaccine? We have to resort to administration of the parasites by intravenous injection. And this can be done. There is a company called Scenarium who work on radiation attenuated Plasmodium falciparum sporozoites, and they have developed a method to generate aseptic, purified, cryopreserved sporozoites of Plasmodium falciparum. And so now we are trying to work with them to get our PbVac also aseptically produced, purified, vialed, and cryopreserved so that we can administer our vaccine by injection, and then we can control the dose that we inject, and hopefully we will be able to inject the necessary dose to attain the 100% blockage of liver infection that we need. The other thing we are working on is on enhancing the immunogenic potential of our Plasmodium berghei platform, because like I said, we inserted Plasmodium falciparum circumsporozoite protein to make PbVac, but we can insert other antigens on Plasmodium berghei, and we can insert antigens from different stages of the Plasmodium falciparum life cycle, for example, or we can insert antigens against other species of parasites, against Plasmodium vivax specifically, which we need a vaccine for. And so we have already in the lab made parasites that express Plasmodium berghei parasites that express Plasmodium vivax CS, and we have even made one parasite that expresses both Plasmodium falciparum and Plasmodium vivax CS, which in theory could be a vaccine against both these parasite species. And we are also working on creating parasites that are self-adjuvanting, meaning that they express a molecule that is an adjuvant of the immune system, and in this way we increase their potency as immunizing agents. This is what we are working on, and this is what I wanted to say. I think I exceeded my time by a little bit, but I hope I was able to show you the main results and where we are at the moment and where we want to go. My final slide is to acknowledge a number of people from various laboratories in various countries with whom we collaborated for this work, and also the funding from the Bill and Melinda Gates Foundation initially, and then from the Malaria Vaccine Initiative, and FCT, a small contribution as well. And basically, I would like to thank you for listening. Sorry that I exceeded my time a little bit, and please do ask any questions that you have. Thank you.
Thank you very much, Miguel, for your fascinating presentation. Now, we open for questions, please.
Come on, guys.
Maybe I can start. It's not exactly one question, but it was fascinating to listen to you, mostly, not also because you have an amazing work, but also because in one part of my academic path, I was at Imperial College, Silut Park, the field station, and they had really a big group in that time working with malaria mosquitoes. So
they still have a very big, yeah.
They are still there. I'm not sure whether
they still have a
beginnings of the 80s.
Research on malaria.
I met Gabriella Gibson, who was working with the mosquitoes, and all the people there. And really, they were feeding the mosquitoes with their own blood.
Mm-hmm.
So yeah. I saw lots of harms like that.
Normally now we do this with, so we can feed the mosquitoes with mouse blood. And the other alternative is to use blood feeders. So it's small devices where we put blood that we get from the National Blood Bank, from Instituto Português do Sangue e da Transplantação, and then we can feed the mosquitoes with that. But yes, mosquito feeding by the researchers has happened a lot in many laboratories. That's true. So and it's not very pleasant to look at.
Okay, yes. So I know that some people want to make questions. I
Please
have
do.
to unmute them. Let's see.
Yes, there are many people complaining they can't ask questions because they have been muted.
Because how can we do that?
The host has to unmute. I'm
But
not sure. Am I a host? If I'm a host, I
yes,
can
you
unmute.
are a host. You
I
are.
am.
Yes, you are.
Okay, so I need to know who. So I'm gonna stop sharing my screen. I may
Yes,
have to share
yes,
it again so that
and
I
you
can see.
see
So
them.
okay, so now I can open the chat, and I have here. I have a question, but I can't unmute. So this is Tomas Werner. Tomas, where are you? We are here. Okay, I have unmuted you.
Hello.
Go ahead, Tomas.
Thanks. Okay,
Connect
so
your camera to Mars. It's much better.
I also
Yes,
can't
please.
do that without permission. Oh, I can. I do have permission now. So let me see. I have a couple of questions.
Sure, go ahead.
And well, one of them is a bit more stupid, and I just thought it was interesting. Is how do you count the mosquito bites? How do you make sure it's 75 mosquito bites?
Okay, so we don't make sure that there's 75 mosquito bites, but we can have a very approximate number. And this is by a very simple method, which is an inspection of the mosquitoes after the bite session, because a mosquito that bites will have blood in its stomach, and that can be seen even with a naked eye, but you see it under the microscope or under a scope, and you can count how many mosquitoes took a blood meal. So what was done was that we would put 75 mosquito bites, sorry, in fact, no, we would put 85 mosquitoes in the cage, because there is a 10% to give a 10% margin. If 10% of them don't bite, we will have 75. And after the biting session, which is 20 minutes, so you hold your arms like this with the cage on each side, you're beaten on both arms. And so there would be 85 mosquitoes, and we would let them bite for 20 minutes, and then we would count the number of mosquitoes that took a blood meal. If it was 75 or more, up to 85, no problem. If it was down to 65, also no problem. We had a 10%, or we had a 10 plus minus margin that was acceptable. If fewer than 65 beat, then we would add more mosquitoes, and we would have a second session to make up until we reach 75. There is obviously a range. It's never 75 sharp, but we would count how many beat by looking at the mosquitoes after the blood meal.
So in theory, every mosquito only bites once?
Very good question. Not necessarily. And in fact, we do what is called interrupted feeding, which means that halfway through the session, it's 10 minutes, then you remove your arms, shake the cage, and put again for another 10 minutes. So this is called interrupted feeding, meaning that the same mosquito may bite more than once. But we consider that each blood meal, a mosquito that took a blood meal corresponds to a bite. There is no way to know how many times they beat. So we know how many took a blood meal, which means that we know that that mosquito had the opportunity to inject sporozoites into the subject.
Okay, thanks.
You had another question, I think.
Yeah, I have one more. It's right from the beginning. It was about the differences between VIVACs and the falciparum. Are the antigens and the proteins between them so different that the falciparum vaccine just wouldn't do anything to VIVACs, like anything at all?
Very good question as well. There are obviously similarities between the two. And it is not impossible that a vaccine against falciparum wouldn't have some degree of protection against VIVACs. The big problem with VIVACs is that experimentally, we don't have the same tools to use VIVACs experimentally as we have falciparum. Let me explain this very briefly so that you understand. We are able in the lab to raise Plasmodium falciparum blood stages. So if you have human blood and the Plasmodium falciparum parasites, you can keep cultures of that in the lab, and then you have the mosquitoes feed on these cultures, and you infect your mosquitoes, and you get sporozoites from your mosquito. So you can get Plasmodium falciparum sporozoites or Plasmodium falciparum infected mosquitoes in the lab with no problem. Unfortunately, this is totally impossible with Vivax. There is no way to culture Plasmodium vivax parasites in the lab. So the only way to get access to Plasmodium vivax infected mosquitoes or Plasmodium vivax sporozoites is actually from the field. People that come to the hospital with a Plasmodium vivax infection, if you collect blood from these people and use this blood to infect mosquitoes, and then you can have access to sporozoites. But this, of course, only in endemic regions where you can get these infections. And in fact, we ourselves are going to receive in 2 weeks' time our first shipment of infected mosquitoes from Ethiopia, infected with Plasmodium vivax, so that we can have Plasmodium vivax sporozoites in our lab. But this is, as you can imagine, very, very different from simply culturing Plasmodium falciparum and infecting our own mosquitoes. So this means, in other words, that to assess the efficacy of a Plasmodium falciparum vaccine against Plasmodium vivax experimentally is not easy. And so there is no definitive answer to your question. My guess is that there will be some degree of cross protection, but it will not be 100% for sure.
Okay, well, thanks.
So Cidalia Pereira also had a question and she needs to be unmuted. Where are you, Cidalia? Can't find you.
Can you hear me?
Yes.
Yes.
Yeah.
Is it Cidalia?
Yeah.
Okay, right. So,
I can't turn my video on. I don't know why.
Okay,
Sorry.
but we can hear you, no problem.
Yeah, my question was about what Tomas said before.
Yeah.
It was about the Plasmodium vivax. If the results you had with this strategy is going into is similar to what is previously seen with the Plasmodium for the other one.
Okay, I'm not sure I understood the question. The question is it that whether we would expect to see this a similar result with Plasmodium vivax? Is that it, or can you clarify?
If the results obtained with the Plasmodium vivax is being into the same results with the others.
Yeah. Okay. So we have made the parasites, the Plasmodium berghei parasites that express the Plasmodium vivax CS. And like I explained in the previous answer, there are a number of things that we did with Plasmodium falciparum preclinically in animal models that we can't do with Plasmodium vivax because we don't have easy access to Plasmodium vivax infected mosquitoes. But we could do a few things. So we already used our parasites, our Plasmodium berghei expressing Plasmodium vivax CS to immunize rodents and then collected the blood. And then we sent this blood to Mexico to a collaborator that has access to Plasmodium vivax sporozoites. And she showed that the antibodies in the blood of these immunized animals recognize and bind to Plasmodium vivax sporozoites that she isolated in her lab. So we know that the vivax equivalent of PbVac is capable of eliciting immune responses against Plasmodium vivax CS, and that these responses are able to recognize Plasmodium vivax sporozoites. To go further than this, we are very limited in the amount of experiments that we can do because of the lack of access to Plasmodium vivax. And that's why we are now getting Plasmodium vivax infected mosquitoes in our lab from Ethiopia so that we can try to do some of the experiments that may help us understand how good this immunization is. So we are very much behind with our PbVac, with our Vivax vaccine compared to falciparum, but we expect that they should behave very similarly in real life.
Okay, yeah, was that about that?
Yeah.
Yeah.
Okay, thank you. So Ana
We have
Paula
another
Sampaio
question.
also has.
Jessica.
Okay, Ana Paula, go ahead.
Can I ask
Sure.
something?
Jessica and Ana Paula Sampaio, one of you.
I am Ana Paula Sampaio.
Okay, Ana Paula, go ahead.
I don't know if you can see me now. Maybe you can.
Yes.
See me. Okay.
Nice background.
So first of all, I want to thank you for this presentation. It is a very beautiful, yes, very beautiful work, and the results are very interesting as
Thank
well.
you.
I would say that decreasing by 95% the parasites in the liver, it's a good achievement. So I would be very happy if I could do something like that. But if you want to increase, and my question is regarding the increasing the percentage of decrease to completely block the entrance of the parasites to the liver. You said that you are going to try two strategies.
Mm-hmm.
One of the strategies is by IV infection. My question is regarding to that IV. Would you think that you would have the same results regarding humoral and cellular responses if you use a different route of infection, because you are using intradermic or something like that by the bite mosquito? And do you think you would have the same by changing the route of infection?
Okay, that's a great question, and it allows me to respond in two ways. First, the question of the issue of the dose, and why do we know, or why do we knew from the beginning that we were vaccinating with a suboptimal dose, and why did we know that? Because in the 70s and 80s, radiation attenuated sporozoites were tested in humans delivered by mosquito bite as a vaccine. But at that time, the regulatory restrictions were not what they were today. And the data from that time, there are papers from the 80s showing that in order to get 100% blockage of infection in the liver, the volunteers had to be immunized by the bites of up to 3,000 mosquitoes. I'm glad I was not one of those volunteers, but this was done. This is published. It was done. And with radiation attenuated sporozoites, you needed between 1,000 and 3,000 mosquito bites in order to get complete protection by Plasmodium falciparum irradiated parasites. We used 4 times 75 mosquito bites. So we knew we were below the optimal dose, but now we are not allowed to do, for ethical reasons, to do 1,000 or 3,000 mosquito bites, which leads us to your question of what is the alternative? The alternative is to inject. And your question is why IV? And this is very interesting because it is clear that it has to be IV. And the reason is because Scenaria, the only company in the world that has the technology and the regulatory authorization to cryopreserve sporozoites for human administration, the first trial they did was precisely by intradermal and intramuscular and subcutaneous. Can't remember. No, it was intradermal and intramuscular actually. And they had 0 protection. And in that paper, which was published in Science in 2011, they were really disappointed when they didn't see protection. And they did a study with non-human primates in which they injected them with intradermal, intramuscular, and intravenous. And then they went to measure the magnitude of the immune responses in these animals, and particularly the liver resident immune responses. And it was a huge difference. It was much higher in the animals that had been immunized by intravenous injection. And so in 2013, Scenaria published this absolutely seminal paper in which they, for the first time ever, showed 100% protection of the volunteers following IV immunization with their Plasmodium falciparum irradiated sporozoites. So nobody knows exactly why, but the truth is that it only works IV. This raises a lot of eyebrows because we are talking about a vaccine for children and infants, and we're talking IV. All I can say about that is that it does work if it's IV. If you talk to Steve Hoffman, who is the CEO of the head of Sanaria, and a great scientist and a very good speaker, and also somebody that will convince you of anything, he will tell you that it is not a problem, and they have now done, and they even recently published another paper where they did IV immunizations of children and infants, and they report that it's almost painless and very easy to perform, and that it can be done safely with no problem. I have never seen it being done. They say and they have done it because it's published. We've seen these results, and and it's the only way that the vaccine works is by IV injection.
Okay, maybe there's something else going on with the bite itself, that it creates some kind of adjuvant effect.
Exactly. That is what several people are looking at. If it works when the mosquito bites and the mosquito bites in the skin, yes, some sporozoites enter directly the vessels, but some will be left in the skin and then make their way into the vessels. So, and it works with a lot of mosquito bites, but it works. So why can't we do it by intravenous or by intradermal or subcutaneous? And there are groups working on trying to enhance the efficacy of those modes of administration, precisely by adding something that will facilitate an adjuvant that will facilitate that mode of injection, which raises less issues than an IV injection.
Yes, because an IV injection in children, it can be done, of course, it can be done, but it's difficult.
It sounds strange, but like I say, they have now done it. Sanaria has done it. There are several papers in which they have shown this. But yes, I understand your concern. It was also my reaction when I first heard about it. But
yes, you need to have trained people to do that.
Exactly. That's what Steve Hoffman keeps saying. It's not difficult if you have qualified person to do that. It is not difficult to do, but you need somebody that knows what they're doing, obviously.
Temos então uma última questão só muito rápida da Jéssica.
Jéssica.
Hello.
Hello.
Thank you so much for your presentation. It was very interesting.
Thank you.
My question is about the volunteers.
Mm-hmm.
A more general question, I think, for the whole subject of vaccination. Since there are very different kind of people with different ages, different ethnies, and the different clinical history, I would like to know how can you assure the same results for the whole population with the results you obtained with your volunteers? How is it done?
Jessica, that's a fantastic question. That's really to the point. It's very interesting because scenarios specifically, they have like any vaccine of this type. The clinical trials start not in endemic regions, but in controlled laboratory conditions, such like we did. So we're talking US or European populations in a controlled setting, healthy people between 18 and 35 years old, no previous history of any disease, no, but there are specific, well, there is a set of inclusion criteria that have to be met for a volunteer to be engaged in this. And so, of course, this is not a representative sample of the world population. And what Scenario have already shown is that the results that you get in US subjects are not exactly the same as what you see in endemic regions in African populations. And this is there is a number of factors that may be involved in this, and you mentioned some of them. It's pre-exposure to the parasite, it's other infections, it's age, it's ethnicity, it's a number of things. And my only answer to you is that, yes, there will be differences between what you see in a phase 2A trial in a European setting or US setting and the real world. People working in this field are aware of these differences. And of course, the idea is to create a vaccine that works for everyone, but particularly for the people in the endemic regions. And of course, the idea is to have a vaccine that is as broad in scope as possible. This may be these differences between US and European populations and then the African populations. We may try to overcome them by, for example, again, tweaking the dose or the immunization schedule, or for example, using different strains of parasite for immunization, because there is not one strain of Plasmodium falciparum, there are multiple strains out there. And so, yes, it's a very good question, because it's something that is not only expected, but that actually has been shown already that it's different. Even immune response is measured in African volunteers versus US volunteers. The magnitude of some of these immune responses, including antibodies and gamma delta T cells, is different. You don't know exactly why, but it is something that has obviously to be borne in mind going forward. But yes, it's very very good question, because we don't, with our 12 volunteers, we don't have a representative sample of the world's population for sure.
Okay, so maybe a future set of volunteers maybe should be with the people from Africa and other more problematic
Sure.
areas.
But this is so any vaccine development is a stepwise process. You have to start with very no, and it's not just somewhere. At the beginning, it has to be really people with no previous history of exposure to malaria, very healthy, 18 to 35 years old. And this is first at phase 2A, this is where you have to go. Then as you move forward, then you can enlarge your sample of volunteers, and then eventually you can actually run your trials in the endemic regions in Africa. And Sanaria are now testing their vaccine in Africa, in the endemic regions, with people naturally exposed to the parasite. But it is a stepwise process that needs to follow these rules, and you cannot jump ahead without having completed the previous step. But thanks a lot. It's really a good question.
Thank you very much.
Thank you.
Muito obrigada. Agora temos mesmo que terminar.
É sério, eu estava a gostar.
Eu tenho a certeza que haveria muitíssimo mais perguntas. Foi ótimo. Agradeço.
Obrigada, Miguel.
Nada. Para mim, eu gostei imenso e tenho pena de não podermos continuar, mas qualquer outra dúvida que tenham, alguma coisa que queiram conversar, alguma coisa, pois a Manuela pode-vos dar o meu e-mail se tiverem alguma pergunta que queiram colocar mais tarde, estejam à vontade, está bem? Sorry, I said this in Portuguese. I was just saying that if there are questions that were left unasked and you would like to ask them at some stage, please ask Manuela for my email and feel free to reach out to me. Just on the subject, make sure that you put Talk Braga, so that I know where you are, because I receive so much spam that I need to know that it's a legit email. So please just write Braga or something on the subject, and then I'll know it's for real. Okay?
Okay. Okay. So thank you very much again, and see you soon sometime, and see the students also soon sometime next week. Okay, bye-bye for now.
Thank you, bye-bye, thank you.
Bye.
Bye-bye, PMG. Bye-bye, alunos do PdVMA.
Ai, eu tenho que parar isso.